Then also you can also derive some features, some properties using amino acids. So, what are the various properties we obtain from amino acid sequences? Amino acid occurrence, that means each time how many times the amino acid occurs. For example, if this is a sequence, so the occurrence of alanine 4, right, this 4. So, what is the composition of alanine? This is equal to normalized by chain length, this is equal to 0.4. So, then you can get the molecular weight. How to get the molecular weight?
Sum of the
for each residue we know the molecular weight, add up, right, Cn of I in W of I minus 18 into n minus 1, right, you can get the molecular weight. Then you can get the residue pair preference, how for each 2 residues, right, dipeptides they come close to each other. For example, here A is with the L, this with A is the T, these are the A is with C and so on, right, you can get the residue pair preference. Then we did the heterogeneity profile. What is heterogeneity profile?
sequence,
Right, you can see the amino acid sequence.
hydrophobicity.
So, here you can see the hydrophobicity, right. So, for each amino acid, for example, this A, T S, L T S, right. So, you can have the values, right, you can get the values and get the profile, this hydrophobicity profile. What is ambiguity characteristics? What is the ambiguity characteristic of alpha helices and beta strands?
The pattern of hydrophilic
Pattern, right. So, for example, the helices we have we can take the 4 residues approximately 10 because of 3.6 residues. So, 2 from one side and 2 from other side. So, 2 are hydrophobic and 2 are the hydrophilic regions, right, that we will see the ambiguity. Right. For example, if you put like this, okay, then these 4 residues, right, they form this. Then for the beta strand you can see alternate behavior, right, you can see the pattern like this, right, fine. So, then we discuss the patterns. So, what is a pattern? How to define a pattern? We can use square bracket, we can use x, right, we can use any specific residue and we can use bracket. What is the meaning of this?
allowed residue.
Allowed, this is the residues are allowed, this means any residue here, particular residue is conserved because you cannot change, here not allowed, this is excluded, right, you can have the patterns. So, what are the PSSM profiles? This is position specific scoring matrices, right, you can see how far each residue is occupied with that same residues or different residues, right. When you align the multiple sequence alignment, from that you can derive this position weight matrix, right, how many amino acids among the how many amino acids for same position, how many times each residue or nucleotide occurs, right. Then normalize with this frequency, then you can convert this into the weight matrix, right. You can what are applications of PSSM? So, you have a wide range of applications because it is based on these scoring matrices. So, you can predict the secondary structures, binding sites, right, aggregating peptides and so on. Right. So, when derive any parameters, right, for any analysis, right, if you deal about the large scale data, right, it is very important to derive data sets. So, in this case we have to do the non-redundant data set. What is non-redundant data set?
less redundancy.
Less redundancy, because if you add the same sequence several times, this introduces a bias. So, in order to avoid the bias, right, we need to take the unique sets, right. So, you can make any cutoff, for example 30%. Then if you see the 2 sequences, right, which are sharing the identity of more than 30%, what do you have to do? You have to keep one or discard the other one, right. So, in this case we need to derive the non-redundant data sets, right, for reducing redundancy. So, what are the various programs we discussed, right, CD hit, blast clust, and PICES, right. Right. So, what is the program we use for the algorithm, right, what is the principle used in CD hit? Clustering, K means clustering. So, you use the different clusters, then you pick up the represent data from each clusters, right. Likewise, we can this works like the clustering techniques. So, then we moved on to the secondary structures, because all these aspects we can do from primary sequences, right. Earlier days the researchers mainly focus on primary sequence, right. After that the applications of the primary sequence data was less because the reliability became less, then moved to secondary section tertiary structures. Now, current scenario again they came back to the primary structure, because now we have the due to the next generation sequencing data. So, we have plenty of data available for the sequences, not for the structures, right. The structural data is still limited. So, you have more number of sequence data. Now, they are deriving various parameters and algorithms to align the sequences, right, getting more attention, right, again in the sequence analysis, right. From the sequence they went the attention to mainly structure, then went back again to the sequencing because of this next generation sequencing techniques. The secondary structures, what are the various secondary structures? Helix, strand and the coil, right, turn coil, right. So, what are the major interactions which are for the secondary structures?
Hydrogen bond.
Hydrogen bonds, right. So, hydrogen bonds between main chain atoms or side chain atoms?
Main chain.
It is mainly main chain atoms, right. Main chain atom. How the helices are found?
II plus.
I and I plus 4 between the NH and CO groups, right. Right. Then we discuss Lorentzian plot. What is Lorentzian plot? Phi and psi, because if you have a dipeptide or any long peptides, so can rotation available is possible in the dihedral angles, like phi and psi angles, that is rotation along n c alpha and c alpha c. So, then if we plot phi and psi, right. So, in this case only some specific positions are allowed to have the specific conformation, for example, alpha helices or the beta strands. You can see the beta strands is very wide and the alpha helices is in narrow range, right. So, you can see these are the allowed positions for the residues to be in the alpha helices or beta strands. Then what is DSSP? Dictionary of secondary structure of proteins. How they derive the secondary structure?
Based on hydrogen and
Based on hydrogen bonding patterns, right, because the hydrogen bonding patterns are known for the alpha helices or the beta strand or turns, right. Use these hydrogen bonding patterns, right. Capstone strand are derived 8 classes, different types of helices. What are different types of helices?
310 helix,
Alpha helices, 310 helix
pi helix.
and pi helix, right. This is the how many turns in alpha helix, how many residues per turn? 3.6 residues, right. So, likewise you can the pi helix and 310 helix 3 and 5, right. So, this is the variation different helices. Likewise 3 strands, beta bridge and the beta strand and coil, turn and bend, right. Fine. So, now from this assignment, so we discussed about the propensity, right. So, how to get the propensity of residues in alpha helix or beta strand? For example, for alpha helices, what is the propensity? Propensity of I equal to number of residues out of I. If it is alpha helix, you put alpha, right, n of I
by total
by n alpha divided by n, right. With 100 residues, right, and 50 residues on helix, right, and alanine, if there are 10 alanine and 7 or 8 are in helix, right, what is the propensity? Point 8 by
1.6.
0.5, this is equal to 1.6. So, is it preferred one or not preferred one?
Preferred.
It is a preferred one, because it is more than 1. So, you can see this is a preferred one. Likewise, you can do it for the all the residues as well as the different secondary structures, right. So, Chavan-Fasman derived this method, that is named after their names, the Chavan-Fasman method. Right. Then we discussed about the secondary structure predictions. And what are the various secondary structure predictions we discussed? For statistical analysis. So, statistical analysis, how we get the secondary structure based on statistical analysis?
Based on Chavan-Fasman propensities.
Right. If you have the 20 residues, they classify in different groups: helix former, strong helix former, right, weak helix former, then helix breaker, helix strong breaker, and then the helix weak breaker. So, based on that we assign values 1 or 0 or 0.5 or -1, and if it is 4, then we consider this nuclear helix. The main aim is there should be at least 4 helix formers, should not more than 1 helix breaker, right. Then we can use the actual values, because we propensity values we have. Based on that, you extend the length. If the value is more than 4, you add the residues, right. If it is less than 4, you cut and then select this segment, right. Then what is information theory? They use information theory, right, to get the secondary structure based on the information regarding central residue as well as 8 residues on both sides, right. GR developed this theory, right. So, there is the information theory. Then heterogeneity profiles, right. If you have the profile, right. So, you can see any patterns, right. For example, for the helix you can this type of pattern, and the strand this type of pattern, or you can have this type of pattern, right, mainly the buried strand, right, and you can see some turn like this, right. Based on the profiles you can see which secondary structures, right, in any particular segment. Then we also use the weighted average, so you see the window length average of 8 residues or 4 residues, and see the peak, and then probably nucleate helix or strand, right. You can various methods to get these profiles. Then multiple sequence alignment, how to get the second structure using multiple sequence alignment. So, you have the aligned sequences, two ways you can do, either in the aligned sequences use Karn method and then use it, or you can use the experimental data, right, whether it belongs to helix or strand. The majority of voting you can get to this residue belongs to helix or strand. Then there are various machine learning algorithms, right, neural networks or support machines, right, they map the input data, mainly the residue, neighboring residues, right, then to predict these secondary structures. Then also we have the consensus method. What is the consensus? Either you can see the meta or the ensemble based method. For example, if you get 10 methods, get the values for the 10 methods, if it is more than 5, then you can say okay, this could be probably helix or strand, or you can get the output of different servers, put into a new server, right, train and then see the output, right. You can also do in these two ways to use this consensus method. Then go tertiary structure. So, what is tertiary structure? Which information we require get from tertiary structure? Coordinates, right. We get the XYZ coordinates, mainly we get the XYZ coordinates, then we get the occupancy and the B factor, right. So, where we get the data for these 3 structures? Protein data bank, it is widely used database, right. You can get the structures. So, what are the various contents in the PDB? Right, you have the header information, name of the protein and the resolution and the secondary structures, sequence, and mainly the XYZ coordinates, right, along with the occupancy and the temperature factors and heteroatoms, right, water molecules and so on. So, there is various tools to visualize these protein structures, right. What are the various tools to visualize the protein structures? PyML is one of the widely used ones, because we can give the high quality figures, right. Also you can do various options, like you can calculate the distance and the bond angle, torsion angle, right. You can use one different models, for example, the space field models and the Cartoon models, right. You can make not like the other programs JMOL and then different other programs are available, right. Rasmol, right, Kings, various other software tools are available, but PyML is widely used, right, for the visualization. Then if you have these proteins, right, then different structure class of proteins. What is the various structure class of proteins? All alpha, all beta, alpha plus beta, alpha beta. What is all alpha? Mainly alpha, all beta is mainly beta, alpha plus beta, we have alpha helix and beta sheets, but they are segregated, alpha beta they are mixed with each other, alternate helix and strand. So, 2 databases called SCOPE. What is SCOPE? Structural classification of proteins, right, and the CATH. What is CATH? Class, architecture, topology and homologous superfamily, right, you can that. So, you can do these 2 databases, this will give you the information regarding classes, folds, right, and families, superfamilies and other things. Then once you have this data, you can derive various features. So, the most important ones and the easiest ones, they give the contact maps, right. What is the meaning of contact map? How to construct contact map? Yeah, if this is the plot connecting these amino acid residues. Right. If these 2 residues, for example, the different residue numbers are in contact, then you put a dot, right. Likewise, you can construct a map, right. If they are in contact, then you can put a dot, we can consider a contact. So, in this case, we require some information. So, to make this contact, first we need a cutoff, which atoms we need to consider. Which atom we usually consider? Either C alpha, you can see C beta, or any heavy atoms, or centroid of any residue, right, that can use. Then the distance cutoff, as per the based on the atom you use, you can use with different distance cutoff, like if it all atoms you can use less distance cutoff for 0.5 to 5 angstrom, and if you take the C alpha atoms or C beta atoms, you take 6 to 10 angstrom, right, you can use it. So, based on these thresholds you can derive this contact map, right. Then this is a 3D representation converted to 2D graphs. You can easily see, right, which residues are close to each other in the 3D structures based on this graph. Then what is solvent accessibility? How much each residue, right, in a protein molecule is accessible to solvent. By rolling water molecule of radius 1.4 angstrom, you can see how much is accessible to this particular solvent molecule. Right, then we classified into different groups. We can see it is buried. What is the meaning of buried? So, for example, if you have a protein, right, some of them are interior core, right, this is called buried, and this is the exposed. So, what is the program used to identify these residues which are displayed, which pictorially represent this buried and exposed residues? AICV, right. AICV can tell you the location of these residues, whether they are in the buried or in the exposed, right. Also the type of these residues and the size, right, gives you the accessibility. Then based on solvent accessible surface area, we did another parameters like reduction, solvent accessibility, buriedness and all. Then based on the contacts, we discussed some of the parameters like contact order, right. What is contact order? This will give you how far the residues which are in close in space, right, and how far they are close in this sequence, right. This mainly was delta Sij, right, normalized by n NDL. So, one is this is the length, this is the number of contacts, right. We can quantify the contacts based on contact order. What long range order? Right, you can use Nij by n, right. So, if Nij equal to 1, if I minus j is less than greater than or equal to 12, right. So, this will account only for the long range contacts, right. Then also we have the hydrophobic contacts, disulfide interactions, cation pi interactions based on the contact between some specific residues, right, whether the carbon atoms or the positive charge and aromatic residues, right, or the cysteine and so on, right. Then we discussed about the superposition of protein structures. What is superposition of protein structures? Align, you can see how much the difference between two structures. It is measured in terms of RMSD. If it is less, that means they are close, right. If it is more, then the structures are not similar, right. Then we discussed about the program, that is the PDB param, right, to calculate all these parameters, right. Then we can discuss about the applications. So, mainly structure prediction, we can do the homology modeling, right. What is the principle used in homology modeling? Right, if the two sequences are similar, so the structures are similar, right. So, there are various steps to do homology modeling. First you get the template, then do the alignment correction and loop modeling, right. You can see the after the alignment correction you do the main chain, can construct main chain, the loop modeling, side chain construction, then optimization and validation, right. You need to do all the steps to do homology modeling. Likewise, you do ab initio modeling and the fold recognition. What is the CASP? Critical assessment, right, for the structure of proteins, right. You can this is a blind test. So, collect the data from the different communities, right, and then see when they publish the data. So, then can compare, right, how far the methods they work is reliable or not. Then you go with the stability. What is the protein stability? This is a free energy change between the folded and unfolded state. It generally is marginal, there is about 5 to 20 kilocalorie per mole, right. So, there are various ways to get the stability with the experiments, right, the differences scattering calorimetry or circular dichromism, right. So, also you can get from the thermal denaturation or denatural denaturation, right. Then if you have these structures, you can get the energetic contributions, hydrophobic, electrostatic, van der Waals, hydrogen bonds, disulfide bonds, right, covalent, the bonded non-bonded interactions, and see how far we can relate this experimental stability, right, obtained from the thermodynamic experiments plus the energetics, right. This is the folded minus unfolded state free energy. So, we found that the hydrophobicity is a major factor, whereas the hydrogen bonds and the other interactions, right, give the shape and then maintain this stability. Then we discussed about the stability. What is the stability? Right, Then we discussed about the stabilizing residues in protein structures. Let us see the some residues, right, which are stabilizing. If you mutate these residues, that will alter the stability and function. Mainly it is based on hydrophobic interactions, long range interactions and plus the conservation score. Then you obtain the data, then compare the experiments, mainly the data from thermodynamic database. What is database for the thermodynamics? Proton database. It has data for the wild type proteins plus mutants. So, we have the mutations, then we can see the important parameters. What are the factors which influence the stability of these proteins, right. For example, the buried mutations, we showed that the hydrophobicity, right, which is a direct relationship with the stability, right. We will get this the direct relationship. In the case of buried mutations, the exposed mutation we get the inverse relationship. What is the inverse hydrophobic effect? So, increase in hydrophobicity decreases stability, right, if we get this type of patterns, right. So, mainly the exposed mutation in the coil region. So, if increase the hydrophobicity, that decreases stability, right. This is the inverse hydrophobic effect. So, this is delta G and this is hydrophobicity, right. Then we discussed about the structure information. We can add the residues which are neighboring occurring within the limit of 8 angstrom to account for the structure information, and also if we have the sequence, right. If it is the central residue, we can get the neighboring residue information to include the sequence information, right. In this case, you can add the sequence information, right, if there are same mutations at different locations, right. Otherwise, you will get the same result if you use the properties, right, because we use the delta p for the difference in property, this is equal to p mutation minus p wild, for example, hydrophobicity, right, difference in values. Likewise, you got the delta g for the mutation wild, right, then you can come these two we relate using correlations, right, correlation coefficient. Then we discuss the folding rate. What is the folding rate? This will tell you the how the protein, right, takes time, whether slow or fast, to fold from the amino acid sequence to the folded 3D structures, right. So, depends, for example, if you see the all alpha proteins which usually fold, right, faster than the all beta proteins, then we account these folding rates based on the structure based parameters, mainly the contact order, long range order, multiple contact index or different parameters. So, we can get these numbers and directly relate to the folding rates, right. There was a direct relationship or inverse relationship, it is inverse relationship, right. If these parameters inversely related with these folding rates, because the more number of contacts, then it slows down the folding process, right. Then we predict the folding rates based on the sequence, right, or from the structures, right, we can predict these folding rates. Then we discuss the protein interactions, right, mainly the protein interactions, protein DNA interactions, protein RNA interactions, right, protein ligand interactions, right. So, if we have the complexes, we can identify the binding sites, right. What are the various ways to identify the binding sites? So, distance based approach, in this case we see the distance between one protein and the other protein or protein and DNA and make any cutoff. If these atoms are within a specific cutoff, we can say they are in interface. Then we get the accessible surface area based approach. In this case, we take the accessible surface area for the whole complex and cut into pieces and take this unbound one, right, and see the difference. If there is a difference in ASA, then we see that they are in the interface, right. Then using then we take the energy based approach. So, we calculate interaction energy, based on interaction energy we can make a cutoff, say 1 kilocalorie per mole. Then if you see it is less than that, we can see this is the interface. Then using this information we identify the binding sites, then from that binding sites we calculate the binding propensity. What is binding propensity? This is a tendency of each residues to be at the interface, right. You can see for 10 alanines on the 8 are in the interface, we can see that they prefer to be at the interface. Then we identify the residues and residue pairs. From that we can see what are the probable interactions. The protein interactions, we discussed the importance of few interactions like electrostatic interactions, cut and by interactions, aromatic interactions, right. Then we can get the importance of specific residues using the binding affinity upon mutation, right. If you can see the free energy change of more than 2 kilocalorie per mole, right, then we term as hotspot residues, right, if it is at least 2 kilocalorie per mole, more than 2 kilocalorie per mole, right. So, where shall we get the information? What is the database? Proximate, right, you can get this database. So, to get the binding affinity upon mutation, you can use this for the understanding the affinity upon mutation. Then the final part, the applications, we discussed about the accommodated drug design. So, what is the protein ligand interactions? So, proteins interact with small molecules, right. Small molecules they trigger these proteins, change the conformation, right. In this case they can be used as a drug like molecule like the lead compounds. So, are there different types of docking? So, rigid docking and flexible docking. In the rigid docking, so protein is fixed, ligand also we can fix and make the interactions, but ligand we can make various configurations, right. With the different conformation you can dock and get this score. For the flexible you can give the conformation changes, right, or the various protein is flexible, right, the ligand is flexible, you can see the probable pose and the type of interactions where you can find between the protein as well as the ligand. But this is time consuming. What are the two different aspects you have to consider in docking? Conformation sampling and the scoring function, right. So, conformation sampling, two types of sampling we discussed. One is the stochastic sampling, one is systematic sampling. Systematic means you have to do systematically, but takes time, right, but you can get one low energy values. But in the stochastic sampling we use various aspects, right. What are the various techniques we discussed? Similarity annealing and genetic algorithm, let us sample, right, the data, different conformation and pick the best. What is scoring function? What are the various types of scoring functions we discussed? Shape complementarity and chemical complementarity, empirical functions, energy contributions and propensity values, knowledge based approach and the consensus. The various ways to get the scoring functions, right, and discuss various software like Glide, Autodoc, right, it's available in the literature, we can use to get the scoring functions as well as to understand the probable pose. Then the virtual screening, so first we need to get the protein, right, we have to model the protein, homology modeling or ab initio modeling, then ligand you have to prepare, then we need to do the docking, so you get the docking score as well as the energy. Based on that then you can identify the probable ligands to be drug like molecules. Then we can also do pharmacophore modeling, so surrounding the protein packet, what is the possibility of the different types of interactions, right, based on the hydrogen donors, hydrogen acceptors, right, or the polar surface area, right, or the molecular weight, you can see how we can model this particular ligand. Then let us see what is QSAR. The quantitative structure activity relationship. The principle behind this QSAR is the activity can be represented as a function of molecular descriptors like the physical properties or chemical properties or two dimensional connectivities or the structural information, right. We can explain the activity in terms of the descriptors, right, physical and electronic properties of these ligands. So, we get the descriptors and finally we relate the structure with the activity and you can measure the performance, right. You can use the correlation coefficient and cross validation techniques, right, to see whether this will fit the model and then use the model for identifying the new compounds, right, by changing the chemical groups. Then we discuss awk programming. What is awk programming? It is a scripting language, it is the pattern driven language, right, data driven language. You can use for systematically use, effectively use for handling the large data sets, right, mainly in the different databases or you can get the output from the different programs, right, you can use it specifically on manipulating PDF files, right, and the extracting data from the many output data. Then we discussed about the different types of algorithms, right, importance of the bioinformatics and classification problems. So, what are the different classification problems we discussed? Right, the DNA binding or not, or the binding proteins or not, transform regions or not, and the driver mutation, passenger mutation, or the malignant or the this benign tumors and so on. So, various steps we had to do. First, we need to take the data set and then feature selection and develop a method and assess the performance and validate. So, these are the various aspects we need to consider when we developing the algorithms. Then we discussed about the applications like classifying the helical and strand proteins, right, mainly we discussed the statistical methods and machine learning techniques and specifically on the applications of VECA, right, which contains various machine learning techniques, right, to classify the different types of proteins and so on. Essentially this course we mainly focused on the algorithms, techniques and the fundamentals and databases, right, and some of the applications on protein folding, stability and as well as interactions. So, we cover the basic aspects as well as the some of the applications. I hope you enjoy these lectures and you understood the aspects of the basics. as well as the for carrying the different projects, like how to approach, right, how to choose a problem, how to approach a specific problem, and what are the very different ways you need to proceed, right, for a successful completion of your project and all, right. So, our best wishes for your bright career, and I hope the program will be useful to you. Thank you very much.